Welcome to Chapter 23. In Chapter 23, we're going to focus on how populations, which a group of individuals of the same species, same place, same time, so really an interbreeding group of organisms, is going to be considered a population. And it's at this population level that evolution will actually occur. So this is going to focus on the differences that we see, such as in the picture here. We can see that these are all the same basic type of clam, but there are variations in terms of their shell design and color. And we're going to look at how we can track these variations amongst the individuals of a population using genetics. Because this change and the fluctuations in the genetics of a population is really the heart of evolution. Over time, these things build up, and that's how we can have these bigger changes. But all that is rooted in these small changes, bit by bit, generation by generation, that occur. So starting off, let's go over some of our terminology. So a population is going to be one group of interbreeding individuals, so essentially one group of a species, but it's not going to be the whole of a species. So I can have a bunch of squirrels in Central Park, but then there's really no squirrels in the city area around there. And so we don't see any more squirrels till we get to another park somewhere else, and then there's a separate group of the same species, but they're not interbreeding because they're not at the same place at the same time. So they don't come in contact with each other. And so this is the key idea. So we can have one species, but because there's multiple populations of that species in many, if not most cases, you can have where each population's doing its own thing as it tries to survive the tears of wherever it's at. Now, gene pool is going to be the entirety of genes that we see in a population typically. And then from that gene pool, we'll have population genetics, which essentially tracks the gene pool and how it fluctuates over time. So sometimes you get new mutations and ta-da, we get a new allele. Sometimes you have something bad happens to individuals and all the individuals with a certain allele die out, so we lose alleles. Sometimes an allele that wasn't very popular might become very popular, and vice versa. And so this will be any type of change whatsoever, but the gene pool is just the entirety. It's, all the stuff. Now, modern synthesis or neo-Darwinism is really just the idea of evolution as it stands today, the idea of natural selection as it stands today. A lot of additional people have gotten research and contributed to this, so we're not using Darwin's stuff exactly. But Darwin's is the basis for what we now have, which we normally refer to as the modern synthesis or neo-Darwinism. And then the last bit, to make sure we're clear, individuals do not evolve. We have fixed genetics. So what happens to individuals is you are selected. So by being selected, you're selected typically to survive, would be one thing possible, or not. That's always the opposite of that coin there. And as far as reproduction. And so when we talk about individuals, we're really talking about, was this an individual of that population that did a better job overall, or did this trait and individuals that had that trait do a better job overall of living to make it past their the onset of their reproductive age at least, and were they capable of successfully reproducing? In some cases, we'll even move on and look at how successful were we at having our offspring themselves live to be reproductive age so they can reproduce. Now the populations, though, they're the ones that evolve. And this is why evolution, too, typically occurs over generations. My generation, the individuals born with me, we have fixed genetics. So we are kind of like a fixed point on this evolutionary pathway. For things to change, which is evolve, evolution, change over time, we need the next generation now, where maybe individuals with red hair, if we want to be mean here, we could say don't reproduce as much, and so we could see that those alleles would become more and more infrequent. So we would see a shift. That's the evolution in action. Now, Hardy-Weinberg. These two guys, Hardy and Weinberg, they ultimately came up with a premise for what it would take to have a population not evolve. Now, you'll see in the next podcast, which will be longer, we're going to focus on essentially the opposite of all of these, because we're going to focus on microevolution. In other words, how things do evolve. But for now, because this works out really nicely for us to figure out kind of what's going on in a population at an exact moment, we're going to focus on Hardy-Weinberg's ideas. And so their basic things were to not evolve, you needed to have a large population. Ideally infinitely large, so there's plenty of extras. You never have to worry about there being, one individual with a certain trait that could just die off randomly. So we want large populations. We want no migration. We want overall everybody just to stay put, so no new alleles can come in by that, new fancy stranger that came in with his, neon blue hair, and now we have a new allele. And we want none of our traits to kind of walk off, where we don't want our individuals that happen to have a certain trait to leave, and now we don't have that trait anymore. We want no net mutations, so overall, we don't want to have anything new crop up or anything get broken by a mutation and kind of disappear. So once again, we're trying to keep things the exact same. We want random mating. Everybody needs to have an equal chance to produce offspring. If this isn't the case, then that means individuals with certain traits will be favored and they will tend to produce offspring more. And so we don't want that. So everybody have to be like, 2 a.m., the bar lets out, and you're just like the next guy I see. That's essentially what we'd have to have for this to work. Doesn't actually happen in nature, but it's what we'd need for this Hardy-Weinberg situation to hold sway. They oftentimes call this Hardy-Weinberg equilibrium because we're trying to keep things as they are. And then no natural selection, so this has to be essentially random survival. You can't have where individuals with certain traits tend to die or live better than others. So this means that no one can essentially be camouflaged or any of that stuff. Predators have to have an equal shot at getting access to and hurting anybody. And so in this way, there should be no change if these 5 things were met. Now in nature, I'm not aware of any population where these 5 things would be met, which is why long term we can't use Hardy-Weinberg's ideas. It doesn't hold up. However, Hardy-Weinberg came up with ideas of how to figure out the exact frequencies of the different phenotypes, genotypes, the allele frequencies. They could basically track the genetics using equations they came up with. And this still works. Even if it's an evolving population, it'll work for that one moment in time. The issue is just it won't stay that way because the population isn't actually at equilibrium. It's going to change. It's evolving. But for that moment, we can get some data in a fairly efficient way, which we can then compare to when we get data over other intervals. And so we can see how a population evolves. So we're taking this idea of a non-evolving population, we're going to use the equation from it, and by applying it multiple times, we can actually track evolution. Seems kind of counterintuitive, but that's how it works. So this is just showing you kind of a picture of this idea here, where it's tracking individuals that are homozygous recessive, individuals that are homozygous dominant, and then the heterozygotes are green. And you'll see these numbers on the bottom for P and Q. I'm going to explain those next. But the idea here is P will always be dominant, Q will always be recessive. So we'll bring this up and I'll try to make this all kind of make sense here as we work out some of the maths involved. This will be one case where we involve math, and you will be expected to know this because this is usually one of the math problems they'll give you with the new AP exam. There's those gridding questions. Expect to see some about this. So I'll do a couple with you here, and then we'll do a lot more in class to make sure we practice so you guys have this down pat. So starting off, here's the 2 equations that they have. P plus Q equals 1.0. This one's going to be a measure of what we will call allele frequency, where P is ultimately the dominant allele and Q is the recessive allele. So this would be like big A, this would be like little a. And so if I say P equals 0.8, what that means is essentially 80% of the total alleles in this population are the dominant allele. Now, this equation says P plus Q equals 1, so that means if A is 0.8, if big A is 0.8, then little a must be 0.2 or 20%. And so together that adds up to 1. So if I know the frequency of the dominant allele, I can then figure out the frequency of the recessive allele, assuming that we're using normal situations where there's only 2 alleles that we have to compare. That's all we're going to do with this. We're not going to look at complex ones where it's, blood typing and all that. But you should know, if I talk about allele frequency, if I say the frequency of the dominant allele, if I say something about alleles, you're going to use this equation, P plus Q equals 1. And always remember to use the decimal version. In your mind, you can think 0.6 is 60%. But for the math, it's going to be 0.6, which means the other one must be 0.4. That way it adds up to 1. If you convert this to percent, that's 100%. Now, over here, we've got not allele frequencies, we have genotype frequencies. So these are going to look at individuals that are going to be homozygous dominant. That's what P squared is. It's homozygous dominant. I'm just going to abbreviate. You've got 2PQ is the heterozygotes. You'll notice you times it by 2 because there's two ways to be a heterozygote. You can be essentially big A, little a, or little a, big A. There's two different ways you can arrive at that, where mom can give you a big A and dad can give you a little a, or mom can give you a little a and dad can give you a big A. So that's why we have to double this. And then we've got Q squared, which will be homozygous recessive. So Q squared, two Qs, which are recessive, and that's essentially just going to be little a. And then this one, of course, big A, big A. So the way this works is it also adds up to 1, which is essentially 100% if we're converting to percents. So it just means if we've got these two alleles only, there's only 3 ways to align them, and if we add those amounts up, we got to get to 1. Now, this one, what you have to be very careful about is Q squared is very informative. That's very useful for us. Me just telling you how many individuals have a dominant phenotype doesn't tell us as much because it's giving us both P squared and 2PQ. Because remember, both of these guys would ultimately have the dominant phenotype. Only the recessive has a phenotype that's unique to its genotype, where we know the recessive phenotype must be Q squared. So if I give you scenarios, you'll see it's fairly frequent that they will either give you P or Q to start a scenario. So if I say something like P is 0.7, then you should in your mind go, all right, well 0.7, that means Q must be 0.3 because those add up to 1. So if you want to, you can add them up to make sure it's 1.0. And once I find those, I can plug them in over here. So if I ask you something like how many are heterozygotes, you just say, all right, well 2 times 0.7 times 0.3. That's 0.21 times 2, so that'd be 0.42. So 42% or 0.42 would be heterozygous. So you should be able to do that. And then the other tricky way I can do things is I can tell you how many individuals are recessive. Or I could say how many have the dominant phenotype, but you wouldn't actually use that number. Let's go through. I'll do as tricky as possible. I'll be as much of a jerk as I can. So if I said something like 84 squirrels have the dominant black fur out of 100. So 84 out of 100 are going to be black furred. And they want to know if the other option is albino. I'm just making simple things here. They ultimately want to know how many individuals are heterozygous in that population. Now looking at this, you should be kind of like freaking out initially, like what the heck. But all you have to do is say, all right, if 84 out of 100 are black, which is the dominant, I can figure out how many are recessive, 16 out of 100, or another way of writing that is just going to be 0.16, will be albino. If they have the albino phenotype, their genotype must be Q squared. So what I've actually figured out now is that Q squared equals 0.16. I can write here. And once I know Q squared is 0.16, I can say, well, Q must be the square root of that, which is 0.4. So now I know Q is 0.4. That means P must be 0.6. And so if I need to solve for 2PQ, it's just 2 times 0.4 times 0.6. Crunch the numbers and you'll get 0.48 and we're done. So it does take more steps, but if you just reason your way through it, figuring out, all right, I either need to know the allele frequency of P or Q, or I need to try to figure out Q squared, the number of individuals that are recessive. Once I get Q squared, simple square root, I've got Q. Once I have Q, I can get P. And once you have Q and P, you can get anything you want. At that point, it's simple. So just make sure that you know these equations and you feel somewhat comfortable using and abusing them. So a couple more examples to make sure that we're all comfy here. So there's a population of squirrels that contains a recessive albinism gene. Sound familiar? If the albino allele frequency is 40%, what percentage of squirrels are heterozygous? All right, that's kind of similar to what I did, but we'll do this one anyway. I'll have to make sure I throw in a homozygous dominant one here or something. So what you need to realize from this is the recessive allele is going to be, because albino is recessive, is going to be Q. Notice it's not Q squared, because I didn't say the recessive phenotype or genotype. I just said the allele. So Q is going to be 0.4 as the decimal version. So that means P must be 0.6. 2PQ is the heterozygotes. That's the way to solve it. So this will be just like the last problem we did. 2 times 0.4 times 0.6. So this will be 0.48 again. Now, if I'd asked you instead for how many individuals are homozygous dominant, you would just say, all right, in this case, homozygous dominant would be p squared. So you could just say, all right, that's going to be 0.6 squared, and that will be 0.36. Solved. And if you want to verify all your work, you can say, well, all right, 0.4 squared would be the q squared. That'd be the recessive phenotype. And so at that point, you get 0.16, if I can write 0.16. And then from there, you can add these up and make sure they equal 1. In this case, they do, so you've done everything right. You're safe. So this is the easier type of problem because I gave you p or q directly. The trick is making sure you don't just write down 40% as q squared or p squared or something else. The trickier type we talked about, in this case, we've got 9 albino squirrels that you count out of 100. So we know 9% of the albino squirrels are albino. Not allele frequency, are albinos. To be an albino squirrel, you have to be q squared. So q squared equals 9 out of 100. If I put that in decimal format, this can trick some people. It's 0.09. Remember, you move the decimal twice. So when it's like 16, yes, 16 gets moved twice to be 0.16. But if it's just 9, you have to move it twice to become 0.09. So now I know q squared. So q is just going to be the square root of 0.09, which will be 0.3. Once I know that, I can get p very easily because it just has to add up to 1. So 0.3 plus what equals 1. I'm going to do this in my head because it's so simple, 0.7. And so if I want to know homozygous dominant now, homozygous dominant is p squared. P is the dominant. I need 2 of them to be homozygous dominant. So that means I will have 0.7 squared. So p squared equals 0.49. Or in other words, 49%. If we're trying to go through, I asked for percentage, so I should follow my own advice here. So 49% of them are going to be homozygous dominant. So this allows us to essentially survey many organisms and just count the number of recessive individuals we see if it's a visible trait. And from that, we can essentially determine the genetic structure of that population. As we said, this will change over time, but it's still incredibly useful to get these snapshots because we can then compare our snapshots and see exactly how things are changing, who's being selected for. Now I'm going to wrap up today just by introing microevolution, and we're going to go through the 5 types of microevolution and talk about some of the last selection-related stuff in the next podcast. But microevolution is just going to be the generational change in genetics in a population. So this means from one generation to the next. You know, what are the shifts where we see maybe a new allele come in, maybe an allele leaves, maybe one becomes more common. So like in this picture scenario, you'll see there's several light-colored moths, and then over time we can see that as generations pass, those guys essentially dwindle away. So this would be microevolution. This would be small changes in the gene pool, in the genetic structure of the organism over several generations. That will be microevolution. We'll discuss the 5 types next time. I'll see you then.